Hi, forget everything that you know about genetics. Today we are focusing on an interesting subject, on a revolutionary field, which is recombinant DNA technology, in short RDT. So, a small modified DNA can open a new door to find solutions for a challenging issues such as drug discovery or disease treatments. Recombinant DNA technology means RDT. R, recombination. The combination of the DNA molecules from different sources leads to produce a new one, which is called recombinant DNA, rDNA. rDNA. So the human gene can bind with bacterial plasmid, bacterial DNA, makes a new one which is called recombinant DNA. And the recombinant DNA allows science to isolate a targeted gene, specific gene, and modified, plus can combine with other DNA molecules which lead to recombinant DNA and makes a new or desired products. So this recombinant DNA can insert it into a living organism or living cells such as E. coli cell in order to produce the desired or the benefit products, or in order to study the structural and functions of targeted genes. So the recombinant DNA, when inserted into a host cell such as E. coli, is called recombinant cell. This is called recombinant cell or cells. So the recombinant cells can produce new products, or we can use it in order to know the structural or functions of our targeted genes. So recombinant DNA technology has a variety of applications, has different applications. Recombinant DNA can be used in medicine to produce a benefit products, or can be used in molecular biology to study the function and structural genes. The targeted gene, as we mentioned it before, can join with plasmids, leads to produce a recombinant DNA. This is a recombinant or new DNA. The new DNA has different applications. For example, in medicine can be used to produce beneficial products or valuable products such as drugs or vaccines. In molecular biology, can be used to study function and structure, or structure and functions of the targeted genes. In other fields, such as in industry can use to produce biofuels. So it has different applications, as we can see in the agriculture also. Now, the most important thing in recombinant DNA is cloning. Let's see what's a cloning and what are the process or what are the requirements of cloning. So recombinant DNA, it means producing a new product through producing or through creating a new DNA. The most important thing in recombinant DNA is cloning. Cloning also is called gene cloning or DNA cloning or molecular cloning. So the idea behind of cloning or the purpose of cloning is multiplication of targeted genes, increasing the number of targeted genes, multiplication of targeted genes, increase the number of genes, increasing the number of our genes. Look, if we isolate a specific DNA, such as insulin gene, this is the insulin gene. We want to isolate this insulin gene. The gene, this one, is isolated gene, which is called insulin gene. The insulin gene located on chromosome 11, which is responsible for producing insulin. And the insulin gene should be inserted into a vector, or a living cells such as E. coli cell. But there is a question, how this gene isolated? This is the first issue. The second issue, how this gene inserted into a targeted cell. And this cell produces insulin products, which is called insulin. But there is a question, how the recombinant cell produces this insulin product. So this issue will be discussed in this video. Now, before discussing these issues, there's a question, what are the main requirements for cloning process? What are the main requirements for cloning process? The main requirements. What are the main requirements for cloning process? What are the main requirements for cloning process? The main requirements are, firstly, the targeted gene, the sequence of gene, and the second one, enzymes. And the third one, vectors. Vectors. And the last one is called host. The last one is called host cell. Let's see the first one, gene. For the targeted gene, we have to have some information. Firstly, you should know the main source of your gene, the source of your gene. And secondly, you should know the length of your gene. And next, you have to know the sequences, the gene sequence. And also, you should know the promoter of your gene. And lastly, you should know the exonic and intronic regions of your gene. Exons and introns. How many exons and introns are available in your gene? So these are some information you have to know, and all the things that we mentioned you can get in a specific website or database which is called National Center, NCBI, National Center Biotechnology Information. All the information are available in NCBI database. And the second requirements for cloning or recombinant DNA is enzyme. Enzymes, in general, in here, we use two different types of enzymes for cloning. The firstly is called recombinant, or is called ligase and endonuclease. Endonuclease enzyme or restriction endonuclease enzyme, endonuclease enzyme, and the second one, ligase enzyme. Endonuclease enzyme, such as, also it's called restriction endonuclease enzyme, for example, EcoR1. Also we have EcoR2, we have EcoR3 and HIN3. The ligase enzyme such as T4 ligase. So what are the main functions of two types of enzymes which are required for cloning or recombinant? As we mentioned it, or we asked the question how we can cut this targeted or insulin gene. Firstly, we have to use restriction endonuclease enzyme in order to cut this one. So restriction endonuclease enzyme can solve this issue. But before, we have to know the restriction sites of this enzyme, of this gene. For each gene, there are a lot of restriction sites. In here also, we have to know the restriction sites. The restriction sites are restriction sites. So when you know the name of restriction site, you can use, based on the restriction sites, you can use a restriction endonuclease enzyme to cut a targeted sequence. After cutting, we have to insert it into a vector before transfer to the living cell. But how? Through using a second enzyme which is called ligase enzyme. The DNA or the targeted gene can be easily binds with the vector or plasmid and can be inserted into in living cell or E. coli cells. So we use different types of enzymes. In cloning, we use endogenous or endonuclease enzyme, restriction endonuclease enzyme, and ligase enzyme. As we said, endonuclease enzyme, for example, HIN3, EcoR1, EcoR2, the types of EcoR1 can be used based on the restriction sites of the plasmid. The enzymes that are used for cutting this sequence of DNA, it should be used the same restriction endonuclease enzyme for cutting the plasmid or vector. And ligase, it works as it acts as a glue. It acts as a glue, molecular glue. Whereas the endonuclease acts as a scissors. The endonuclease enzyme firstly recognize these sequences, recognizing this one, and secondly, this is the first, secondly, it cuts or cleaves at the specific site, which is cleavage site or restriction sites. And also the restriction sites cut, some of the restriction sites produce cohesive ends. For example, look at this. Look at these sequences: G, A, G, A, T, C. This is our sequence. The complementary sequence, it should be palindromic. C, T, A, G. This is the palindromic sequence. So, the restriction endonuclease enzyme cuts this one. How? Because it can be read from right to left or from left to right is the same. Just like a word of madame. Madame. In here, madame. If you read it in this side also, you can read madame. GAATTC. GAATTC. So the restriction endonuclease enzyme cuts at the first two nucleotides, then it moves to here. Look how it works. This is called palindromic sequence and can be recognized easily by the endonuclease enzyme. So the third requirement for cloning, or can be used in recombinant DNA, is called vectors. Vectors. In general, when we use bacterial cell, we already use plasmids as a vector. So plasmids are extracellular chromosome or extracellular DNA. Extracellular DNA. This is a plasmid. It's a circular in shape, double-stranded DNA, and it has or it contains some of the genes that are not or that cannot be seen on chromosomal bacterial chromosome. This is called plasmid. Plasmid or vector. If we use bacterial cell, we have to use plasmid as a vector for transformation, for inserting our gene into a host cell. The typical plasmid should contain the following points, or should has the following features. The firstly, it should contain the ORIC, or origin of replication. ORIC, ORI, it means origin of replication. So the plasmid, one of the features of the plasmid, it can replicate itself without DNA replication, without bacterial duplication, without cell division, because it has ORI, it has origin of replication. So also it contains the second thing which require for gene expression is called promoters. The polymerase enzyme can recognize promoter or promoters on the plasmid in order to start for replication and gene expression. So this is a promoter sequence. Promoter sequences. If I ask a question, why PCR not or cannot be used for cloning? We use plasmid and bacterial cell for cloning because the PCR is a machine. It leads to produce or multiply our targeted genes only. It's in vitro technique. But the gene cannot be expressed. The protein not produce it. Whereas if we use plasmid, yes, the protein can be produced by those genes because the plasmid contains promoter. It has a promoter for gene expression. The third feature of the plasmid is called multiple cloning sequence. Multiple cloning sequence. Multiple cloning sequence and multiple cloning sequence. Multiple cloning sequence required for what? For inserting our gene. This is our targeted gene. This is our DNA sequence. DNA sequence or target gene or gene sequence. We want to insert it into a plasmid, so there's a place, and also it has a or two cleavage sites, cleavage site or restriction sites. This is a cleavage site, that the restriction endonuclease enzyme can recognize these two sites easily and cut it. Then we can insert our gene sequence into MCS, multiple cloning site. So this is the second issue. The third issue, which is related to the last requirements, main requirement for cloning process. Firstly, we said genes, then you have to know the name of the enzymes and vectors. This is the vector. We took plasmid as a vector for inserted our DNA sequence into a host. So the last requirement is called host cell. Host cell. Host cells. We use different types of cells for cloning, for expression of our targeted gene. In general, based on the research, around 56% they use mammalian cell. cells for cloning. And the second one, 24% they use E. coli cells, bacterial cells. E. coli cells. And also, there is another cell which is called insect cells. Also, we have two other cells which is called yeast cells and also animal and plant cells. So around 30% they use yeast cells, or others insect cells, or they use animal and plant cells for cloning. But the most cell which used for cloning, which is called mammalian cell or E. coli cells. These are the main requirements for cloning process. Finally, in short, we can say that the main steps for cloning, it includes three main steps. The first one, you have to select the targeted gene, target sequence, target gene. After selection of target gene, then you have to increase the number of targeted gene. How you can increase the number of targeted gene? By inserted into a host cell. And lastly, after inserting into a host cell, we have to extract, the final step, we have to extract the product, the protein. So, extract the proteins. Extraction, the desired product, These are the main steps for doing recombinant DNA or for doing cloning. In the next, we try to focus on how we can extract the products or the desired product for targeted sequence.